Hey guys, it's Emba Jamir. It's my second video on respiration, and it's the continuation of the last video. I hope you guys are doing good and great. So we're gonna learn today what is Krebs cycle, what is the electron transport chain, and what is chemiosmosis. Before that, we gotta hop into the structure of one of these mitochondrions right here. So this is the outer membrane of the mitochondrion. This is the double membranal structure, the mitochondrion, and this is the outer membrane. This is the inner membrane, as you guys can see. The space, the peach color space between these membranes is called the intermembranal space. Now, these inner membrane, they are foldings, as you guys can see here, foldings, and these foldings are called cristae. They're present to increase the surface area of the mitochondria, the inner membrane. And the spaces between these inner membranes are called the matrix. Now always remember that this space is called the matrix. We're gonna know more about it. Now here what happens is the Krebs cycle totally occurs in the matrix. And what happens in the Krebs cycle is that for every molecule of glucose that occurs in that happens that undergoes glycolysis, forms into pyruvate. We know that we learned that the pyruvate forms. When pyruvate forms, it converts into acetyl-CoA in the link reaction. Now, two pyruvate will form, so two acetyl-CoA will also be formed in the link reaction. Now, the acetyl-CoA is basically the first reactant of the Krebs cycle, and we're gonna see here. See, this is the acetyl coenzyme, right? So there are gonna be two of them because two of them were produced. So remember, all of the Krebs cycle occurs twice. So whatever products will be formed here will also be formed twice, right? Okay. So firstly, what happens? This two-carbon compound acetyl coenzyme A forms citrate. Now, when this happens, when it forms citrate, it's a six-carbon compound. We're not going to learn how it converted into citrate because this is not a part of our syllabus, but just know that the first step is always acetyl coenzyme A converting into citrate. Now, the citrate releases a carbon, and due to the release of carbon, it forms an intermediate compound, which is a five-carbon compound because it loses one carbon, so five compound. And this carbon that it loses, it combines with oxygen to form carbon dioxide. So basically, the NAD here, what happens when carbon is released? When carbon is released, hydrogen is also released with the carbon. And when hydrogen is released, the NAD it gains the hydrogen and becomes reduced. Now, this intermediate compound here, which was produced, it forms oxaloacetate. Oxaloacetate is a four-carbon compound. And what happens here is that it loses another carbon, and this carbon goes and attaches to um, oxygen again to form carbon dioxide. So basically, two, and two carbon dioxides are released right now, and this all of this happens twice, so four carbon dioxides are released. And there are a lot of steps that happen in between this third step, intermediate compound and oxaloacetate. We're not gonna discuss them because they're kind of complicated and they're not included in our syllabus. We just gotta know that how many reduced NAD and how many reduced FAD are formed, and there's also a ADP being converted into ATP here. So as I told you guys, it's gonna occur two times. So ATP to two ATP produced, two NAD reduced produced, two reduced FAD produced, and as a result, when there's two here, two here, two here, so six reduced NAD and two reduced FAD, two ATP, and four carbon dioxide are formed. We can see here, and the end product of Krebs cycle is always oxaloacetate. And we can also see here that 2 ATP, 6 NAD reduced, and when there's NADH, which it means it's NAD reduced because it has gained the hydrogen. So, and 2 FADH2 produced, right? Now, in the next step, we're going to learn the last step of aerobic respiration, which is oxidative phosphorylation. Now, oxidative phosphorylation, it consists of two parts, which is electron transport chain and chemiosmosis, right? And it always occurs in the inner mitochondrial membrane. Always remember this, that it occurs in the inner mitochondrial membrane. Okay, how does it occur? This is the electron transport chain right here. You guys can see these five electron transport proteins. What happens is this NAD that comes from the Krebs cycle and the FADH2, they split up. Due to the split up of these electron hydrogen carriers, hydrogen protons are released. And due to the release of hydrogen protons, electrons are also released. Now these electrons, they what happen is that they go into the electron transport proteins. When this happens, the electron transport proteins, these electrons, they move in from one electron transport protein to another. Now this movement of electron transport protein to go in from one to another and another from to another, this releases energy, which we learned yesterday was chemiosmosis, that release of energy due to the movement of um, sorry, wait. So chemiosmosis is gonna be related to forward, but this is right here that the when the energy is released due to the electrons moving here, this energy is used by the hydrogen protons here to be pumped into the inner intermembranal space. Now this is the matrix where they're already present, and it uses energy. This energy that is released by the electrons, it is used by the hydrogen protons to go into the intermembranal space from the matrix. Now this happens by facilitated diffusion. These protons go into the intermembranal space by facilitated diffusion. Now when this happens, the proton gradient here increases, and it becomes a lot of, too many hydrogen protons get surrounded here, so the proton gradient increases. Due to this, the hydrogen protons, they go into this ATP synthase enzyme that is present on the intermembranal space. Now when they go into this ATP synthase enzyme, they release energy. And due to this release of energy, ADP combines with the inorganic phosphate to form ATP, which we learned yesterday that in chemiosmosis what happens is that energy is released due to the movement of hydrogen ions across the gradient to form ATP. So this is how it's produced. This is chemiosmosis. What is chemiosmosis? The movement of hydrogen protons across the membrane to release energy for ATP to be formed. So yeah. And what happens here is that when they go into the ATP synthase, they are transported back into the matrix. Now, when they're transported back into the matrix, they're gonna go back again, and they're gonna go transported into the intermembranal space. How to intermembranal space? Now, how to stop this reaction? So there's always oxygen present here as a final electron acceptor. So it accepts these protons and it combines with these protons to form H2O. So always remember that oxygen is the final electron acceptor, and without oxygen, the electron transport chain cannot continue. So yep, that was it for today, guys. Hope you guys understood the electron transport chain, Krebs cycle, and the chemiosmosis process and the structure of the mitochondrion. Thanks so much, and see you guys tomorrow or whenever I'm gonna make a video. Till then, bye bye.